Well, welcome. I am Joanne Geer. I'm the Executive Director for the Westchester Biotech Project, and we want to welcome you to our consortium on translational research in the microbiome. We meet monthly, and each month is a really wonderful topic. And today we're going to be talking about antibiotic selection related to cystic fibrosis. You'll hear from George Tetz, and you'll be hearing from him in a moment, but first we want to tell you a little bit about the consortium and about ourselves. To take the next slide. Keith, would you like to say a few words about the consortium and yourself?
Yeah, I'd like to welcome everyone and just mention ILSI, the Institute for Life Science Entrepreneurship. We're one of the founders of this consortium of scientists on a translation of the microbiome. We started several years ago and have been stalled like many people around the world due to COVID, but making a slow comeback. And I think we're going to be hitting our stride this year and having an annual meeting to begin with and to continue with this workshop. So I'm really happy to be here and to represent ILSI and to continue to learn about the type of research that's being done in the microbiome to really bring it closer to the clinic and into the world of products. So thank you all for being here.
Adela?
Hi, everyone. Thanks for coming. We are happy to have you here. We have an exciting discussion about antibiotic selection in cystic fibrosis. And we like to bring people together and gather around Joanne enthusiasm, which has always attracted me, and creating a community. We are all part of it, and foster innovation, because I think microbiome is it's new and require the development of many techniques, require lots of hands on board, and because of that we need to be together and understand the journey of a microbiome based product from a concept to the market. And I think today I'm going to shortly introduce our speaker, but I think Joanne is going to add a little bit more about Westchester Biotech, right?
Yes, thank you, Adela. Westchester Biotech Project was launched in 2017 with the idea of bringing together researchers, data scientists, and engineers, and those who love them. And we have gathered many times similar to today and been listening. What are some gaps and pain points that can have a real impact on helping to grow the life science community broadly and also for individuals. And I really love this, tomorrow is today, which is actually a quote from Martin Luther King that, you know, we really need to be doing today what we want to see tomorrow. So we have several sort of main themes, which you can see here. Career Consortium, which looks at workforce development issues, but also individual career paths, and clinical research is a big one that we're developing. It's a wonderful career. It's a wonderful enhancement to a career, and just a really fantastic field for people who may not ever get an MD or PhD, but can really have an impact on tomorrow's discoveries. We're working on our data analytics hub within which this consortium resides. And Blueprint 2030 is kind of a cheeky thing for a little organization like us to lead, but we didn't see anybody around us really making big plans for our community, so we decided to step up. And we actually announced that in 2019. So we've been chipping away at it, and you'll be hearing more about that soon. We can't do it alone. We work with lots of wonderful partners. Working with ILSA, with Keith has been fantastic, and with Adela and her companies. And we're Montero Language Services, I'll just point out, because they have made a 2-year commitment to us that's really allowing our programs to grow. And they are experts in translation and localization for elite documents like intellectual property and so on in the life sciences. So they're really wonderful partners. We've got a lot of events coming up. I'm not going to go over these right now. They're all on the website, but you're welcome to get involved with any of them. We are often looking for speakers for monthly series. So if you have an idea about something you'd like to share, we'd certainly love to hear it. And there you go, Adela.
Thank you, Joanne. And I'm going to introduce today our speaker. His name is George Tetz, and he's an MD, a PhD. He is a CEO and a CLL at CLLA Therapeutic in the Human Microbiology Institute. And he comes from a rich, what attracted me when we invite George is because his background, it's very rich. He comes from a academic, industrial, and non-profit organization. And throughout his career, he has focused on applying leading-edge genomic approach to a variety of medical problems. He has been in a background of numerous groundbreaking studies in the field of medicine and microbiology. And he aimed at the discovery of the new regulation pathways linking gut to microbiome and neurodegenerative disease. Author of 50 patents in the field of microbiology and drug development. Prior joining CLA Lab Therapeutics, George has worked in numerous collaborative drug discovery programs and contribute to bringing more than 18 novel candidates in the clinical development. He has over 18 years experience in the microbiological research and drug discovery, and he is coming today to talk about his newest research, which is focused in microbiota, including bacteria and bacterial viruses, and implicated in a variety of multifactorial disease. His latest work on antibiotic resistance in mixed biofilms has contributed to the development of a novel principle for antibiotic selection. And today is going to apply that for cystic fibrosis patients, which they face a big challenge, for example, infections. Welcome, George, and we look forward for your questions. And we will bring our attendees at the end in our discussion, and it's going to be an interactive stage with George and his a new diagnostic technique. Thank you, George, and welcome.
Yes. Thank you very much. Thank you for this introduction. My name is George Tetz. I am MD, indeed, PhD in medical microbiology, and is a co-founder for non-profit Human Microbiology Institute. We are New York City-based organization, scientific research organization, and we also do a lot of collaboration. And specifically, we collaborate on this project with a diagnostic-oriented company named TGV-Dx that is located in Buffalo, but also within New York State. So thank you very much for this introduction. It's my absolute pleasure being here and presenting today. So we're fighting antibiotic resistance with a novel culture-based diagnostic test named ADB-Finder. And ADB-Finder, it utilizes a novel microbiome-based strategy for antibiotic selection. And this strategy enables us to select antibiotics effective even against infections caused by multi-drug resistant bacteria, and to do it within just 3 hours. And I would like everyone to take a look at these astonishing numbers. The survival of patients with different infections, such as pulmonary, urinary, or skin and soft tissue infections, rely on timely selective. Sorry, excuse me. Really apologize for that. Because for some reason, everything is jumping today. Excuse me. Sorry about that. Okay, now it's good. The timely selection of antibiotics for these infections is critical for patients with these diseases. And well, let's take a look because if we're talking about the ways how we select infections, how we select antibiotics, there is a number of gaps, and in all today's culture-based tests in the view how to select antibiotics. So I'm going to start with a statement that literally, as of today, one of the major problems for the ineffective antibiotics are not only related to the spread of antibiotic resistance per se. I'm going to make a statement that, as of today, one of the reasons why antibiotic therapy fails is because the conventional methods for antibiotic selection simply cannot select the effective antibiotics. And let's take a look. Let's start with a brief introduction why that happens. So as of today, the literally vast majority of all culture-based tests, they select antibiotics against the lead pathogen that are studied in a very artificial settings, when the lead pathogen is isolated from the biosample, and after that, its sensitivity to different antibiotics is conducted. So in other words, the most important part here is that the antibiotic selection is conducted against the leading or predominant pathogen when it's cultured as a monopathogen, is pure bacterial culture. However, at the site of infection, the situation is totally different, because at the site of infection, the lead pathogen is surrounded by dozens of different microorganisms, and all these other bacteria significantly impact on their antibiotic sensitivity of these lead pathogen. So let's take a look what is missed by conventional tests, and I will try to guide you why it is so important. So the first part is so-called collective antibiotic resistance. Sorry. So, excuse me, what does it mean? So microorganisms at the site of infection live in a very tiny community in the form of microbial biofilms. And within this microbial community, different bacteria can produce different factors that can inhibit antibiotics. And these factors can protect the whole bacterial community. So in other words, for example, the lead pathogen that per se is sensitive for the beta-lactam antibiotics, it is protected against these beta-lactam antibiotics by beta-lactamases that can be produced by bacteria that surround it. And this part is totally missed by conventional tests. Another important part that is also missed by standard methods is that microorganisms at the site of infection form microbial biofilms. And within these microbial biofilms, these microorganisms, they're covered by additional surface membrane. They have a lot of extracellular polymeric matrix. And all these make microorganisms much less sensitive for the antibiotics. So it's not really the antibiotic resistance, but it's more like tolerability of microorganisms against antibiotics. And the existence of microbial biofilms is not recaptured by standard methods, and significantly decreases the accuracy of the antibiotic selection tests. So the third part is that, of course, microorganisms that surround lead pathogens modulate the expression of antibiotic resistance genes. So the expression of antibiotic resistance genes of the lead pathogen at the site of infection is not the same as when it's grown as a monopathogen culture. And moreover, the number of genetic tests, they're also unable to address this question, because the presence of antibiotic resistance genes does not mean that this factor is expressed. So that is why, for one of the reasons why the culture-based tests are still to be on the priority list for the selection of antibiotics. And the second important factor is because the lead pathogen very frequently rely on their presence of so-called bacterial helpers that help the lead pathogen to withstand antibiotic assault by modulation of the type of their growth. And finally, last but not the least, is that very often when antibiotics eliminate or decrease the abundance of the predominant pathogen, other microorganisms that are present at the site of infection can take the lead and can significantly promote their growth, causing the second tier of infection. And this scenario is particularly critical and well seen in patients who are immunocompromised. In other words, microorganisms that are believed to be non-pathogenic, like, for example, E. coli, can contribute to a severe disease in this case. And this is just another reiterate the summary for the previous slide, showing that with ATB Finder, we're going to model environment at the site of infection, and that this environment is not captured by a standard and conventional antimicrobial susceptibility test. ATB Finder is a paradigm shift in how we select antibiotics. It's 2 or 3 40-well plates. Each well is filled with a novel host mimicking nutrient medium named TGV, developed by Record. And this media provides it has 3 major unique features. First, it provides the growth of literally all microorganisms, even very fastidious, from the site of infection. Second, these microorganisms are grown in the form of mixed biofilm. And the third one is that the visible bacterial growth on the well starts very quickly, within just 4 hours post-plating of the material. The nutrient medium in each well is supplemented with the antibiotic or with antibiotics mix based on their therapeutic guidelines for the treatment of certain disease. And the antibiotics are added, and this is a little bit tricky. So they're added at the concentrations that can be practically achievable at the site of infection based on the pharmacokinetics or pharmacodynamic parameter of the certain antibiotics. So we're not using the minimal inhibitory concentration. We're not selecting the antibiotic sensitivity. We help the doctor to address the question which antibiotic are effective or not by applying antibiotics to the whole microbial community in their biosample. And we can run simultaneously up to 138 different antibiotics and their combinations. And we're able to provide the answer within 4 hours that are at least 44 hours faster than any other available conventional antimicrobial susceptibility testing, specifically culture-based. The workflow is very straightforward. The biosample is plated directly without isolation of pure bacterial culture to the wells, to the nutrient on the top of the nutrient agar. And after that, the plate is incubated for 4 hours at 37 or can be adjusted. And after that, the presence of bacterial growth is visualized and the antibiotics are categorized as effective or ineffective. Effective antibiotics are suggested to be those that can completely kill all the microorganisms or cause the stasis of microbial growth. In other words, from the wells where there is no bacterial growth. And the ineffective antibiotics are suggested to be those in the wells in which the presence of microbial growth can be seen. So here's a real-life example of the performance, of the microbial growth in the wells, and it's clearly seen the presence or absence of microbial growth. Our primary indications are those with unmet medical need, or specifically those in patients with pulmonary, skin and soft tissue, and urinary infections. So the list is shown here. And we're currently starting the second tier of clinical trials in patients with cystic fibrosis, including those infected with Burkholderia cepacia. The third tier would be the clinical trials in patients with ventilator-associated pneumonia, the infection, which are very frequently caused by multi-drug-resistant bacteria, and in which the time is of essence, because it's always well recognized that it's critical for many diseases to put the patient to the right antibiotic as quickly as possible, because the increase of the duration of the time where the patient is not receiving an appropriate antibiotic is very frequently leads to, unfortunately, either to the complications or to the mortality, what is also frequently seen. We conducted numerous in vitro studies, and I will just very briefly guide you through some of those. We studied the overall specificity and sensitivity, negative predictive values, positive predictive values of the ATB Finder in various settings, and it was shown to be highly accurate. And addressing the question about the microbiota, we conducted the number of metagenomic analysis when we analyzed the microorganisms that give growth on the different medium So we compare TGV medium with the other standard medium like Columbia agar or nutrition agar or LB agar, for example, figuring out what is the diversity of microorganisms that can be grown on TGV versus versus the variety of different microorganisms that are present at the site of infection. And this table clearly shows that the TGV enables the cultivation of literally all microorganisms from the site of infection, and that this number is strikingly higher than that can give growth on a standard medium. But this part, of course, it was theory, because it was our original concept that it should be working. However, in order to address, is this, approach applicable to the real life, we conducted the human pilot study in 35 patients with cystic fibrosis. Cystic fibrosis, it's the most frequent genetic disorder that results in the formation of a very sticky mucus. And these patients, specifically children, they're getting chronically infected with multiple microorganisms. So unfortunately, their lungs can be viewed as an incubator for the formation of multi-drug resistant bacteria. These patients are treated for life with multiple antibiotic regimens, with both inhaled and IV or oral antibiotics. Unfortunately, the median survival is around maybe 45 years, maybe 46 right now, due to the appearance of Trikafta. And they have a dramatic decrease in the lung function, specifically due to the chronic infection that is impossible, unfortunately, to stop. So we conducted a prospective open-label study in 35 patients with cystic fibrosis, and we compared the performance of antibiotics selected with ADB Finder. We monitored them for 2 years versus 2 years prior time period when these patients were treated with antibiotics selected with conventional antimicrobial susceptibility tests. So in other words, each patient was used as its own control. And I would like just to reiterate that cystic fibrosis is a very representative disease because these patients have multiple infections through their life and each year, and I will be showing how the landscape was changed. So first thing that we noted that the number of hospitalizations of these patients due to pulmonary exacerbations was dramatically reduced. So at the time when the antibiotics were selected with the standard methods, the total number of antibiotic of hospitalizations throughout the year across these 35 patients was around 45. So in other words, literally all the patients had to be administered 1.2 times to the hospital. However, that can be seen on the left-hand side. However, once antibiotic therapy was switched to those selected with ADB Finder, the total number of hospitalizations during the first year dropped down to 2, and at the second year, we were able to completely arrest the hospitalizations due to pulmonary exacerbations. And on the right-hand side, you can see the same data, but more like on the individual scale to see how patients actually performed on these antibiotics. Another critical parameter in these patients is forced expiratory volume. That reflects the lung capacity, and which is known to be highly dependent on the presence of infectious process in their lungs. And totally, we received a very good result. So there was over 20% increase in the forced expiratory volume across the all patients. However, when we took patients who at their baseline had severe or very severe abnormal forced expiratory volume, so it was the mean number was was 32 or 33% from the predicted normal. The FEV1 was dramatically increased. So there was over 27% increase, and I'm talking right now about not the relative percentage, but about the percentage points So there was a dramatic increase in the lung function, and you can see that all patients responded beautifully to these novel antibiotic regimens selected with ADB Finder. But the cream of the crop is the eradication of Pseudomonas aeruginosa. So patients with cystic fibrosis, as I mentioned, they have multiple microorganisms. So their lung microbiota is really rich, it's very rich. However, one of the most crucial pathogens is Pseudomonas aeruginosa, specifically multi-drug resistant Pseudomonas aeruginosa. And initially at the baseline, 33 patients out of 35 had chronic Pseudomonas aeruginosa infection. And once antibiotics were switched from the standard methods to those selected with ADB Finder, this number was reduced by 81%. And it's absolute miracle because once established, Pseudomonas aeruginosa is believed to be impossible to be eradicated in these patients. And next, we analyzed, well, what happened? Maybe we were giving them too many antibiotics, you know. However, the situation looks totally different. So if you'll take a look on the left-hand side, the total number of systemic courses a year prior to the use of antibiotics selected with ADB Finder across patients with Pseudomonas aeruginosa infection was 70. However, after we switched to the antibiotics selected with ADB Finder, this number dropped down to 45. And on the right-hand side, you can see the individual heat map of the antibiotics selected with the standard AST versus those selected with ADB Finder. So you can see there each patient is divided into two columns. At the left-hand side, you can see the antibiotics suggested to be effective with the standard AST, marked with their blue squares. And on the right-hand side, the antibiotics selected with ADB Finder. So the blue antibiotics are those that were used by the physician for the treatment, and the crosses are those antibiotics that were found to be effective, but they were not prescribed. So one can see that literally ADB Finder was selecting different antibiotics compared with the standard methods. And at the end, we also analyzed the total number of all systemic antibiotic courses across all the patients during 3-year time period before the antibiotics were used with were selected with ADB Finder versus 3-year post during the selection of antibiotics with ADB Finder. And we were able to reduce the total number of systemic antibiotics by 2-fold. And it's a great victory also, just because looking through the lens of the antibiotic stewardship, we're just reducing the number of systemic antibiotic exposure for these patients. If to summarize these results, with ADB Finder, we're able to put the patient to the right antibiotic the same day, saving up to 96 hours versus standard methods. And ADB Finder is able to select effective antibiotics in literally 100% of all cases when conventional antimicrobial susceptibility tests are failed to do so. I would like definitely to thank the team from TGV-Dx, Sir Yakov Kogan, who is the CEO of the company, and of course the team from Children's National Hospital and from the University of Buffalo, and Professor Victor Tetz, who work on this project. So this is just the outline and the summary of ADB Finder. With ADB Finder, we're able to select antibiotics to treat infections caused by multi-drug resistant bacteria. We're able to find antibiotics that can eradicate the pathogen. We can avoid the antibiotic antagonism. And it's interesting, it's a quiz for me as a scientist, because this phenomenon is very well known, but the reason for it seems to be, pending and elusive. But the problem is that very often when you take two antibiotics that both are active against these pathogens, when you combine them together, they stop working. So this is named antibiotic antagonism. It can be very easily found in the Petri dish, but what's more important, this phenomenon is also very well known for the frontline physicians. So the remaining points are that we're able to test up to 130 antibiotics within the same patient. The antibiotic therapy results in a twofold decrease in the total number of antibiotics exposure. And finally, and this is, I think, also important, is that we're able to select more sometimes affordable antibiotics on the one hand, but on the other hand, we're able sometimes to select antibiotics that are not so restrictive, playing another role in the prevention of the antibiotic resistance spread. So thank you very much for your attention, and I would be happy to address any questions. Thank you.